I just wanted to give you a short movie about some of the PDFs that we have on the Exam 2 review. These PDFs are ones that I had drafted before from earlier classes and I use for lecture notes. So they're pretty similar to what you see in on the board when we go up through there. This one, for example, is from September 24th's class. It's talking about how we can have a weak threshold or a weak stimulus or a strong stimulus help to activate a neuron. So I'm just going to circle the little arrow around here, um, the cursor indicating this is sort of the transduction site of a sensory neuron, and then it progresses down an axon to the terminal itself. The main idea is that you transduce your signal here. This depicts several different options here. One is that it's a weak subthreshold stimulus that's being presented to the neuron. And in this particular case, what happens is that you have a depolarization, as depicted here, but it's not sufficient enough to actually exceed the threshold of minus 60, so therefore you don't get an action potential generated down the length of the axon. And so you don't have any release of neurotransmitter either. If you have a weak stimulus, then but stronger than what you had when it was subthreshold, what will happen is that you get your depolarization. You can exceed threshold, which is depicted by this dotted line here, and then you can actually have action potentials emerge from the top of that. And then what this means then is that as it's propagated down to the next part of the axon, you can see that there's many action potentials that are being generated. And as is noted here, the amplitude or magnitude of these action potentials is the same as if you had a really strong stimulus, which is what is shown down here. So this is a depiction of a graded membrane potential from a really strong stimulus. Here is where it's actually, we're having a lot of firing as a result of that right at the initial segment, which is right up there. And then finally, this is when we have a lot of high frequency firing from the action potential. And so the main idea is basically that a stronger stimulus will give you a higher frequency of action potentials. A weaker stimulus will give you a lower frequency of action potentials, and that's one way that we can encode intensity of the stimulus. So that's what we have there. The next slide talks about this connection that we have for a Golgi tendon organ. And so this is depicting a Golgi tendon organ with a lot of tension on the tendon and how that can come back, feed into an inhibitory interneuron, which then can feed to an alpha motor neuron and help slow down the frequency of action potentials. So we had a similar picture drawn in class. And so the main idea is that this inhibitory reflex here makes it so that you shut down the activity of this alpha motor neuron. And so that was on 926, I believe. Maybe it's 929. Let's check here if I have it up. It was 929 was that day where we drew that out. So you can check your notes there. But this is the main idea, so how it's kind of going through this cycle. The stretch reflex, this one we saw a lot more. We talked about a lot more, and how we have our muscle spindle reflex right here. Our muscle spindle is present, and it's a sensory organ that can then pick up stretch or load on our larger muscle itself, and that in turn gets propagated back, that signal is propagated back to the spinal cord through an interneuron and then out through a lower motor neuron. And so this is a case where if you activate this sensory organ here, it'll cause a stronger contraction to occur here. And these steps and sequences here are similar to the ones that we talked about in class. Finally, then we have these. And we talked a lot about the corticospinal tract. Here's the upper motor neurons that reside up in the motor cortex. These project down and cross over at the level of the medulla, and then synapse onto alpha motor neurons. And those alpha or lower motor neurons then project out to cause contraction of this extrafusal muscle fibers. And so this is just showing the kind of movement of information from the cortex down to it. The other thing that's depicted here that's kind of interesting is that, if we cut off the upper motor neurons here, and we just allow the lower motor neurons to persist like we have right down here that I'm circling down here. What's interesting is if we leave this and let it sit there, it's still hooked up to sensory motor neurons. And so you can have a reflex occur even in somebody who has a lesion up in upper motor neurons. So you have a patient, for example, that wouldn't necessarily be able to induce a voluntary motion from because they've had a lesion of their upper motor neurons, but they are able to cause a reflex pathway to be activated. So, and so this kind of compares those two different types of scenarios. The sensory thing was depicted here, but really it's described on the next slide, where you can see two different pathways that we talked about. This is anterior, but it's actually the lateral spinal thalamic tract, the one that we have to know. And it just shows the pathway of these different, how these neurons are traveling up to the spinal cord.